Our next new fellow is Dr. Philip Hugenholtz. Philip is a microbiologist who has made landmark contributions to the understanding of uncultured microbial diversity, evolution, and ecology. Please make him welcome, Philip.
Thank you, and I'd like to thank the Academy also for welcoming me into the fold, and I'll endeavour in the next years to make some useful contributions. So I'll begin by apologising to the astrophysicists about co-opting their term dark matter, but hopefully in the next 12 minutes I'll show you that there are some valid parallels. So after Darwin presented his thesis about that all life is united by a common ancestry, there were many efforts to synthesize this, normally taking the form of a tree, and this is a well-known one by Ernst Haeckel back in 1866. And you know, if you're a researcher back in those times, naturally looking around you, plants and animals take a very high role in your view, and they should be the large branches within this tree, and of course crowned by humans, the crowning achievement of evolution. However, there are also single-cell organisms were known by this stage, thanks to the microscope, for a couple of hundred years already, and these were relegated to the base of the tree, which by looking at they're the least morphologically informative, and so they were all lumped together. And actually that situation didn't change much for the next 100 years. Roger Stanier, a very famous microbiologist, sort of threw his hands up in hopelessness back in the 60s and said, you know, we can only really tell apart a few basic types, but beyond that, the relationships of these organisms to each other is a complete mystery. Well, that all changed in the following decade, largely thanks to this man here, Carl Woese. He was influenced by Linus Pauling's papers just saying that, you know, now that we know that DNA is the blueprints for life, maybe we should be looking at that as the basis for working out the evolutionary relationships between organisms. And he took that to heart, and he targeted the ribosome, the protein synthesizing machinery in our cells, which is universal amongst all cellular life forms. And he looked specifically at the ribosomal parts of the ribosome, which are very conserved. He actually started with the smallest one here, the 5S, but he moved on to the middle-sized one, the 16S RNA, and it has a number of features that make it a very good evolutionary marker. And so he started to sequence that, or back in those days make fragment profiles of them in order to compare them to each other, in order to see how organisms were related to each other. And it turns out that the Haeckel tree is upside down. So instead of the microorganisms occupying this small part of the tree, it's the other way around. All of the large things, animals and plants, occupy a small part of the total ribosomal RNA tree. Now, when you think about this for a few minutes, it makes perfect sense, because plants and animals are relative latecomers in evolution. So therefore, you would expect them to occupy a smaller part of the overall tree. So what it pointed to is this vast amount of diversity that's solely occupied by the microbial world. So to make this even, compound this even further, compound's probably not the right word, is that that tree that you saw there is mostly organisms that you can grow on an agar plate. That's mostly how we've learned about microbiology in the last couple of hundred years, is that you grow them on an agar plate to bring them into our spatial realm so we can look at them. So here's an agar plate. Now somebody's put their hand on it, and you can see these organisms growing up. But if you count the number of these colonies on the plate and you compare them to the number of cells that you see under the microscope, there's a big disconnect. So that disconnect is typically, like if you're looking in soil, that disconnect can be 1,000-fold. If you're looking in the human gut, it can be less, but still the majority of organisms don't appear to be growing into colonies, and this was termed the Great Plate Count Anomaly, identified in the mid-80s. So Norm Pace, who was a colleague of Carl Woese, got around this cultivation bottleneck by applying that ribosomal RNA. PCR, Joseph introduced earlier, thank you. He had been invented in the 80s, and so Norm Pace said, well, what happens if we just extract the DNA directly from an environmental sample, and then we use PCR to amplify up that ribosomal RNA gene, and then we can compare them to each other, and we've got a marker of what organisms are present in the sample. I was fortunate enough to be in the PACE lab in the mid-90s doing a postdoc, and I was looking at a hot spring in Yellowstone National Park, applying exactly these methods to that ecosystem. And I remember being literally blown away by the fact that so much of the diversity in that 16S was not represented by cultures. So this is a tree I made 20 years ago, and it's showing major lines of descent within the bacterial domain. And the blue lines indicate major lineages that have one or more cultivated representatives, and the red ones, ones that are entirely composed of uncultured organisms. So the hot spring I was looking at is OP, and so all these OP numbers were sequences that were coming initially from that hot spring. So most microbial diversity is unexplored. Another way to, and this is 20 years ago, where we are today, there's about 100 of these lineages, of which about half don't have cultured representatives, so-called candidate phyla. Another way of looking at this is to look at the phylogenetic diversity that's in the tree and put it on a y-axis here in units, and then look over the years, and we look at the 16S sequences that are in the public databases. And so in the dark green, these sequences from cultured organisms, and you can see there's been a steady tick over the years between 2000 and 2012. And in the green are these sequences that are fished out of environmental samples. And so back in 2000, only 15% of the database was coming from these environmental uncultured organisms, and the situation basically reversed by 2012. So all of this grey stuff here is so-called microbial dark matter, all these organisms in our biosphere that we know very little about. So the question would be, how do we get to the genomes of these organisms and fast? So if we want to know more about these organisms, if we can't grow them readily, the quickest way to find out more about them is to sequence their genomes. So the answer to this is essentially just a simple modification of what Norm Pace put forward back in the 80s and 90s, where instead of just sequencing that one marker gene, the 16S RNA, you just sequence all of the DNA. So if you take isolates, and give an isolate, that's genomics, and if you sequence an environmental sample directly, that's called metagenomics or ecogenomics. So metagenome analysis has been evolving very, very rapidly, and it's been driven primarily by this huge uptick in sequencing throughput that Justin alluded to. So back in the early days of metagenomic analysis, 13 or 14 years ago, you would sequence millions of bases of DNA from a sample, and really your best bet at that point was to just look at the genes that are present on those individual fragments to work out what's going on in your ecosystem. These days, you're getting out billions of bases at a go, and you can do a lot more with that. If you have that many more pieces of DNA, you can assemble them into larger pieces, and in fact today, we can assemble them into high-quality genomes directly from the environment, which we call population genomes or genomes from metagenomes. And once you have that, then you can work out the metabolic reconstruction to work out what they're able to do. And so population genomes are actually starting to outpace genomes that we're getting from isolates. There's one thing I wanted to point out. Oh yeah, I want to tell you one little tiny anecdote here. So an original study back in the early 2000s, funded by the Department of Energy, that generated about 62 million bases of DNA, cost $500,000. And on today's instruments, we can sequence that for less than $5. So that's just to give you an idea of the scope and the change in the sequencing technology. So there was a commentary a couple of years back talking about this technology that's coming along and enabling us to add genomic foliage to the tree of life. So metagenomics, which I've just described, but also there's been rapid advances in being able to take single cells out of a sample and then sequence that single cell directly. So we've got these great, powerful tools for being able to rapidly fill in the tree of life. So we've taken these data and we've invested a fair bit of time making a standardized genome-based classification. Now, why might you do that? Classification is really a human-made construct, but then so is time. We've petitioned time into a hierarchical classification system so that we all know to be in this place at this time to listen to this talk. So it's a useful human-made construct. And we've attempted to do that, but use the the genome-based tree as the structure on which to put that classification. So what we've done is we've taken the standard 7 hierarchical ranks, and we've overlaid them onto a tree, accounting for differences in relative evolutionary divergence. So we feel this is probably the closest to a naturalistic classification. And from doing this, currently the release 78 from GenBank had there's 83,000 genomes in this classification, and we've had to make at least one or more changes to 84% of the taxa to make it consistent. So what can you do with this? Well, one example is to look at the human gut microbiome, and so here we have each dot represents one of these population genomes, and if it's red, it's named. That means it's got a cultured representative, and if it's blue, it's unnamed. So on this axis, there were 250 people that contributed to these data, and so this tells us how prevalent a particular genome or population is in the human gut, and on this axis is its relative abundance. So what you can see immediately is that about 70% of these species haven't been named. So 70% of our gut bacteria across the human population have not been characterized because they haven't been cultured. And that's important because the human gut, or the gut microbiome, has been implicated in many physical and behavioral problems within our population. So we need to be able to communicate at this level first. You can look at some of these very large uncultured groups, these candidate phyla. So we did that with a talented PhD student, Yunkit Yeo, and we looked at this thing that was just called candidate phylum TM6, originally identified as a 16S sequence in peat bog in Germany, and we were able to retrieve about a dozen of population genomes representing this group, and they were quite distinctive in that they were very reduced genomes. They lacked a lot of the normal apparatus that you would see in free-living organisms, and they also had the potential or genes which allowed these organisms to parasitize the ATP or energy pool of a host. So we could conclude that this was a parasitic lineage, and if you made a phylogenetic tree of these markers, it would indicate that parasitism is actually ancestral within this lineage. Another recent example, the work of Rochelle Su, was to make use of the genome tree to look at the evolution of oxygenic photosynthesis and aerobic respiration in cyanobacteria, which is a highly contested issue in the field. So the first thing to notice from this tree is that this group here, the oxyphotobacteria, represents all of the cultured representatives of the cyanobacteria. And then there were two main basal groups, which have been named Melanobacteria and Cerecytochromatia. And these groups are only represented by uncultured organisms. So this provided us with an outgroup in order to look at the early evolution of photosynthesis. And what we immediately noticed that none of the 50 or so population genomes that we have representing these groups have any skerrick of photosynthetic apparatus, suggesting that photosynthesis arose relatively late in the cyanobacteria, just on the oxyphotobacteria lineage. And then if you look at the respiration in these groups, you'll also notice that it evolved independently in the different groups, supporting that late evolution of photosynthesis. I notice I'm at the end now, so I need to jump. So it's a very achievable target to get these additional genomes, which will help us fully describe the microbial world. And with that, I'll just thank my team. Thank you.
Philip, any questions? Take a minute or two for a question. Yes, up the back. Any other questions?
Mark Westerby, Macquarie Uni. Thanks, Phil. Could I push a bit further on this matter of being uncultured? So if you take, for instance, the human gut situation, presumably it's fairly easy to produce the conditions under which these organisms would grow if you put your mind to it. Is it just that people haven't gotten around to it, or is the reason to believe there's something terribly difficult to culture about all of these?
I think it's a bit of both. So yeah, the politically correct way to describe it is as yet uncultured. meaning that there's the scope for culturing. So I think you have a bit of both of those. So typically, in the past, cultivation has been a little bit just put it at aerobic under normal atmospheric conditions, grow it for a couple of days, put it on an agar gel and see what you get. Now that's pretty crude. You can do a lot, as you mentioned, you could reduce your oxygen content, grow it anaerobically. You could put it on a gelling media that's not toxic to most organisms. That was only discovered a year or two ago. And you can definitely make the conditions better. But I would say, I mean, that's empirical data that we have. We have 1,800 population genomes, so we know what fraction have cultured representatives and what don't, and it's about 30% have representatives, the other 70 don't. So there are a lot of people now, because the human gut microbiome is becoming a hot area, that are claiming that we can get most of them. So now, if you have a decent classification system, you can actually empirically test that. So I would say it's going to be a bit of both. We will see that number go up, but I would guess it's going to asymptote, and you're not going to get the whole lot.
One final question from Joseph.
Thank you. It's fascinating. My question is about, in my field, the single-cell genomic technologies are really rewriting biology and how we understand how the organs operate. So how much this single-cell, single organism, or even single-molecule sequencing you are using, instead of growing, pretty much isolating these organisms and sequencing them individually, and rather than meshing them all up and reconstructing their genomes?
Yeah, so there are definitely pros and cons between single-cell and metagenome-based methods. And in fact, you go to conferences and there's two teams and they're like in combat, but in fact they're complementary. So the pros of the single-cell is that you're getting a single cell, and so you're looking at not a hybrid of a population, and so you can connect all of your SNPs, for instance. The downsides is with the current technology, because you have to amplify the DNA in that single cell from femtograms up to something that we can sequence, you get a huge bias in the coverage, and the typical completeness estimate of a single cell is 30 to 35%. So you're missing a chunk. But those techniques are rapidly being enhanced. There's new enzymes to replace the standard amplification ones. So I expect that technology's in a real growth phase, and it's going to be very improved. So I think the combination of the two is, the other thing is it's a lot more technically demanding than just going and sequencing a chunk of poo and then putting back the genomes from it.
Thank you. Thanks, Philip.
Thank you.
Thank you very much.